Got it. Hello, everyone. So it's your second day. So what is PivoWorks? Right answer. We have everyone should be here or on Zoom, except one class, autonomous underwater vehicles. They have a schedule conflict because the only time the MIT swimming pool is available is during lunchtime. So that's where they are. So with that, it is great pleasure to have our first speaker for the seminar series, Professor Ariel Furst. And her title of the talk is Taking Inspiration from Nature, Bio-Inspired Systems for Sustainability and Clean Energy. What I would like to tell you is that when we do the feedback forms at the end of the program, one of the things the students mention is that how much they enjoyed the seminar series, right? Many of the talks are the kind of talks that you may not actually, it's little maybe slightly different than what your interest might be. But then after we hear so many talks, you're going to realize what a wonderful world out there in engineering and science, right? She's, so just very short introduction. She is the Cook Career Development Professor of Chemical Engineering at MIT. She got a PhD at Caltech, and she did a postdoctoral fellowship at UC Berkeley. And what I liked about her bio the most is she is passionate about STEM outreach and increasing participation of underrepresented groups in engineering. So let's welcome Professor Furst.
Thank you. Excellent. Okay, so this is the first one of these seminars. So I've talked to some of you a little bit beforehand. I'm not scary. If anything is confusing or you have questions, interrupt me. It's much more fun than sitting here for an hour, I promise you. Okay? Just so everybody knows, my labs are in building 56, which is on the other side of campus. But if anybody's interested in seeing what the chemical engineering labs look like, we can figure out a way to get you a tour of our labs this summer. And yeah, so I'm going to talk about a couple different stories today. But I wanted to talk about what motivates all of our research, because we work on very different things in my lab. So this is a picture of Earth at night. Does anybody notice anything? Excellent, right? Where we see electricity is in developed nations. In what's often called the global south or developing nations, electricity is scarce. In fact, 1 in 10 people globally don't have access to electricity. And that means about 2.5 billion people rely on dirty fuels to heat their homes and cook their food. And the problem with this is that using those types of fuels actually has direct impacts on human health. So we all know electricity is pretty green, right? It doesn't create any visible dust or anything like that. When we think about alternatives like burning wood, we normally do that outside, right? Because it generates a lot of smoke. If you're inhaling that smoke constantly from things like coal or wood, it actually has a direct impact on your health. So we can see that in energy impoverished areas, those are just regions that have limited electricity, there are higher rates of things like asthma and heart disease. And so that means that lack of access to energy technologies can actually hurt your health. So in my group, we try to develop technologies to achieve energy equity and justice. And this is the idea that in general, industrialization has harmed disenfranchised people the most. And in order to right those wrongs, we not only need to make technologies that are accessible to everyone, but we also need to remediate the harms we've already caused. And so in my lab, we try to make technologies, and I'll talk about a couple different ones today, for equitable healthcare, sustainable materials, and materials that actually allow us to remediate environmental harm. So before we get into it, you probably saw bio-inspired on my first slide. That's a very fancy word for looking at nature, realizing it's had about 4 billion years on us for evolution, and taking inspiration from that rather than starting from square one. So I'm sure many of you are familiar with this building. It's actually based on a deep sea sponge that is very light and actually hollow, but is very structurally rigid because it has to withstand very high pressures in the deep sea. For centuries, Humans have been taking inspiration from animals such as the pangolin for things like suits of armor for protection. And then the last one is my favorite, and we'll talk about it a little bit more later. But does anybody know what this is, what the red is? Yeah, it's Velcro. Excellent. So this is synthetic Velcro, and this is what inspired it. This is a burr containing a seed, and this is animal fur. So the concept of Velcro is basically designed based on how seeds stick to animals so they can be transported. So in my lab, we take inspiration from nature. Who here has heard of redox processes? A few of you? Excellent. Okay, more hands. Redox is oxidation and reduction, right? All we're talking about is movement of electrons. So pretty much every biological process is founded on the movement of electrons. And so what my group does is we use materials engineering, bioconjugation chemistry, which is just a fancy word for sticking things together in a way that doesn't hurt biomolecules, and synthetic biology, which is another fancy word for saying we take DNA we want in a cell, we force it into a microbe, and then we make the microbe do what we want it to do, to characterize electron transfer in biological systems. So I'm going to tell a couple stories today. The first one is about human health, the second about sustainability, or the second about environmental remediation, and the third about sustainability. So the first is about human health, and I just wanted to show a picture of some of the people working on this project. This is Song Yi, a postdoc, Marjan, another postdoc in the group, and Cindy, a third-year grad student. So who here has heard of HPV? And I'm going to keep asking questions, so the faster you guys answer. Good, excellent. Who here knew that it caused 98% of cervical cancer? Awesome. So this is a virus that causes cancer. We have a vaccine for it, but most people don't have access to the vaccine yet. And so what happens if you're infected with HPV is that your body can normally clear an infection, but there are certain strains that are high risk that generally turn into cancer. So as we can see on the left here, in developing countries, as you would expect based on the story I've told so far, there are both much higher rates of HPV infection and mortalities due to that infection. Much of this is due to lack of diagnostics. So if you can't diagnose an HPV infection, you don't know whether or not it's cleared, right? So the way we do this in developed nations is a pap smear. You go to your doctor, they take a swab of cervical cells, and they look at them under a microscope. That's fine, it's somewhat subjective, but it's even worse in developing nations. There, they take a swab of acetic acid, swab it on your cervix, and look for lesions. Doesn't feel very accurate, right? So the majority of lesions are missed with this, and it's a painful and invasive process, so many people don't want to do it. So how can we think about diagnosing HPV equitably? And what does it have to do with gold-plated chicken wings that Kim Kardashian has been standing for? So my postdoc Marjan looked at food like this and said, if gold leaf is inexpensive enough to put on food, it's probably inexpensive enough to make diagnostics out of. And so she developed a workflow, which we'll go into more in a minute, that allows us to take a patient sample and electrochemically diagnose whether or not that patient has high-risk HPV in half an hour. It costs us about $2 to do this assay. So the overall workflow is you have a patient sample, in this case, it's a cervical swab. We can amplify the genetic material, and I promise you, we'll go through all this in more detail. So if you're completely lost right now, just give me 5 minutes. We can then use that to activate a protein that will cut up DNA on an electrode surface. So this is our actual readout. If our protein cuts the DNA, our signal turns off from black to red. If our DNA is left alone because there's no virus, our signal stays on. So does that make sense to everyone? If DNA is cut, signal turns off. If DNA is not cut, it stays on. So what do these electrodes look like? Well, remember I mentioned that edible gold leaf? Well, we can make electrodes out of it. We put these on transparency film, which for those of you, probably everyone in this room, they used to use this to teach. They would write on overhead projectors on transparency film. Has anybody seen or heard of this before? Awesome, like 3 of you. Excellent. This is how I know I'm getting old. So we put this on transparency film, which looks like a normal 8.5 by 11 sheet of paper. It's just clear. We make the gold leaf into stickers, and we physically take those stickers off and place them on this sheet. This costs us about 50 cents to make in the lab, and it's only about 16 cents in gold. That's about 10 times less than any commercial electrode right now. And those commercial electrodes have other problems. So here's some fancy imaging we did. So these are just two different commercial brands of disposable electrodes and our electrodes. And you can see here just some different types of imaging. And all I want you to take away from this is that these two are very rough, right? They look really inconsistent, while ours kind of look like waves. They're very neat and regular, and they kind of undulate. And we can see that here in these micron scale images, where basically what this is telling us is that we have really high quality gold. So our gold is not only 10 times less expensive, but much higher quality. And what this translates to is we can actually detect proteins better. So what we're looking at now is just cutting that DNA on the electrode. So if we see, so here's 1, that means no signal change. 0 means we've cut all the DNA. So decreased signal is what we're looking for. We see here on these commercial electrodes, no matter what the concentration of that protein, we really have trouble detecting it. Here we can get some at low concentrations, but it's really only with our electrodes that we can get concentrations of detection across a broad spectrum, right? So what this means is that our electrodes are not only higher quality gold, but they allow us to do more efficient biosensing. And these are both critical for this assay, because without both of these, none of these other steps would matter. So now let's talk about that protein. So now we're here. How do we actually specifically turn on a protein? Has anyone here heard of Cas12a, Cas9? Excellent. So you're generally familiar with the concept here, which is basically that if we have this protein loaded with a specific sequence of what's called guide RNA, so we can just program that in to whatever sequence we want. In this case, we make it complementary to our target DNA. Basically, what happens is when that binds, in Cas12a, it'll cut either single-stranded or double-stranded DNA. Or sorry, Cas12a. So we see that here. So that means if we have our single-stranded DNA on our electrode, if that Cas12a is activated, that DNA will cut. Make sense? That's how we turn our signal off. And we get very high specificity for this. So HPV16 and HPV18 are two different strains of high-risk HPV. So those are two of the ones that cause cancer. What we see here is if we load that guide RNA with a sequence for HPV18, we only see a signal decrease for HPV18. And if we load that Cas12 with 16 guide RNA, we only see a signal decrease for 16. That means we can distinguish between strains of HPV here with the same sensitivity as PCR. So that's great, but HPV infections often have very low viral loads. So we might need to actually amplify our signal, right, in order to actually detect it. That's where LAMP comes in. So sure, everyone here has taken a COVID test. That's great, but they use thermal cyclers that cost hundreds of thousands of dollars. That's why you have to send your sample to a core facility where they run hundreds of these a day, right? Is there a way we can do this for not $100,000? Well, we can if we use this one specific type of amplification called LAMP. LAMP is an isothermal amplification method, which just means it runs at a single temperature. So we can amplify. This is just showing it makes these dumbbell structures. All that you need to care about here is that we get these things called amplicons that are different sizes, but that all contain our specific region of interest. And we can do it with a resistive heater under our electrode that costs about $10 instead of a $100,000 thermal cycler. So we can amplify our DNA. And some of you might be wondering, well, if PCR is that expensive, why isn't everybody using LAMP for all these diagnostics? Well, the problem is when you do LAMP, you can get false positives. So the good thing about isothermal amplification is it amplifies a lot, so it makes a lot of DNA. The bad thing is it can make a lot of DNA whether or not it's your target. So we can see here, you see these HPV18 that it's positive for. have specific banding patterns. That means that it's the correct amplification. If we look over here, these are all false positives, which we can tell by gel because there's different banding patterns, right? Just the pattern of the band sizes looks different. The problem is, if we use a standard assay for this, it's going to turn whatever color we want it to turn, whether or not it's the correct sequence. It's only when we throw this into our electrochemical assay that we get specificity. So what we've been able to do here is remove false positives. That make sense? Awesome. So just went through a lot of biology, but just to summarize, if we have HPV in the sample, we amplify it. That will activate a protein to cut our DNA on our electrode and turn our signal off. Yes? Excellent. It works in clinical samples. So what we have here are 10 clinical samples, as well as a no template control. That's our negative control. And what we see here is we have 100% sensitivity and 98% specificity. We have one false positive out of all of these readings for HPV, high-risk HPV from patient samples. And so what we're doing now is integrating this into a microfluidic device so that you can take a vaginal swab at home, run this on the device, read it with something that looks like a glucose meter, and detect HPV infections in your house without having to leave. The idea here being that if we can make things like this, it allows people to use telehealth more effectively, thereby enabling more equitable access to healthcare because you don't have to travel to a clinic to do these tests. So we'll just pause here. Does anybody have questions about this story? Yeah. Yeah, so basically PCR normally varies in temperature, right? So PCR to amplify, you go to high temperature and then low temperature and then high temperature and low temperature, and you need a fancy piece of equipment for that. LAMP, instead of just using your normal set of one forward and one reverse primer, and I can talk more about what all that is later on, you use a set of 6 primers so that once you start that amplification process, that dumbbell structure basically self-amplifies. So it makes this weird structure that you don't normally get with PCR and allows it to self-amplify at a single temperature. Does that make sense? Kind of, yeah.
I guess we have a question online, so maybe we can try this. Anula Jakula, would you like to ask your question?
Yeah, so I think I missed this. How was the high false positive rate dealt with?
Yeah, so the high false positive is just with LAMP because LAMP will amplify off-target things as well. However, because we have the Cas12 that has a specific sequence, even if we have a ton of amplified material that's the wrong sequence, it won't activate the Cas12. So we only get activation if that LAMP worked properly. So we can take those LAMP materials and stick them in our assay and only detect the real HPV cases.
Thank you.
It just means making more of it. So usually with things like PCR, the way you detect it is by generating more small pieces of DNA with the same sequence. So with LAMP, you can either generate the proper sequence, or because it does this kind of self-amplification, you can get lots of DNA that doesn't have the right sequence. But we can specifically detect the DNA with the right sequence. So it helps us limit or decrease our limit of detection because we get more material to detect from. That's a great question. So you notice how it said gel negative and gel positive? We actually ran that on PCR, which is the most sensitive method, and that actually had a low viral load. So that actually wasn't a false positive. It was a real positive. We're just more sensitive than gel, which is the standard way to look at amplification. That's a good question. A false negative, yes. It's with LAMP, basically, if the enzyme is not active, you wouldn't see amplification and we wouldn't detect anything. The great thing about the chips that we're making that I showed in the very last slide is that we can incorporate a positive and a negative control. So we'll be able to tell that the chip is functioning properly and that the assay is functioning properly, and then whether or not you're HPV positive. Ariel, could you repeat the question so that people? Oh, yes. Yep. That's a great question. So the question is, how does the Cas12a result in a signal off? So what I didn't go into at all is we can put something that gives us an electrochemical signal on the end of our DNA. So when it's tethered to the electrode, it gives us a signal. When it's cut, we wash the electrode and then it goes away. And so we turn our signal off physically by just removing the redox active molecule. Awesome. Next story. We got 3 to get through. So this next work was done by a postdoc in my lab, Amruta Karbalkar, and an undergrad who started with us as a freshman, Rachel Almark, who is now a senior. So highly recommend undergraduate research. Has anyone here heard of organophosphate pesticides? No? That's probably good, because basically they have the same mechanism of action as chemical warfare agents like sarin gas. And that's why they work to kill insects. They're about 50% of the pesticides used globally. They're used more in developing nations than in developed countries. And the problem with that is that what you see here is dispersal of them, and everybody's wearing PPE, right? Personal protective equipment, goggles, masks, everything like that. Most people who are responsible for distributing this don't have access to that. So what that means is they're breathing this in for years and years. And although we know what happens when someone gets an acute poisoning, that means they have enough to have an immediate physical reaction. What we're only finding out now is that long-term exposure can cause respiratory diseases, infertility, and birth defects in the children of people exposed to this. And for those of you who have looked at chemical structures before, you can probably see the similarity between things like serins, soman, and cyclosarin gases, and paraoxin and parathion, which are the pesticides we're talking about. And the specific thing here is what's called this organophosphate linkage. It just means that there's this organic, these carbon-containing molecules bound to this oxygen on this phosphate group. So that's here and here. And this is basically why it can target your nerve muscles or your nerve endings in your muscles. And so this can cause acute paralysis or those long-term health effects.
We have a question.
Yeah.
Is Agent Orange one of those pesticides?
I'm actually not sure. I believe so. I think it's sarin gas. Yeah. So nasty stuff. The great thing is nature has evolved enzymes that can degrade these into benign compounds. So there are enzymes, which is just a fancy word for proteins that can do some sort of activity, that can degrade these OP pesticides, so that's the organophosphates, that's these guys, to this P-nitrophenol, or as I'll call it, PNP, which is not harmful to us. It's also yellow in color, so we can detect it by just UVVis absorbance, so just looking at its absorbance spectrum. And the enzyme that can do this is called organophosphate hydrolase, or OPH. And so what it can do is it can take this compound and break it down to that compound. Make sense? That's fantastic, right? We could, in theory, use this to protect our farm workers, all that great stuff. The problem is proteins are very delicate. We heat them up, if we expose them to too much UV light, if we expose them to too high or too low temperatures, or too high salinity, or too low salinity, or too high pH, or too low pH, they degrade. That's not great, right? So what we figured out in my group is that we can actually tell cells to put this protein on their surface. So now we just tell those cells to have a specific linker sequence. So instead of just making a lot of this protein inside the cell, it makes a lot of the protein on the surface. And what that means is now we can take those cells and use the whole cells to degrade those compounds. What's even cooler is we found that if we take these cells and freeze dry them, so anybody who's had like astronaut ice cream, anything like that, we can do that to cells. The cells are dead, but the proteins are still active. So we have really, really, really cheap biomaterials that are very stable. So we can now take these freeze-dried cells and use those to degrade the organophosphate pesticides. And what we see when we use those cells is 100% efficiency for degradation. So we can see here that based on the input of the pesticide we put in, this is the concentration of pesticide, we can detect the equivalent amount of that degradation product. That's great, right? Well, this is UVVis absorbance spectroscopy. And what we see here is our limit of detection is about 5 nanomolar or 5 micromolar at best, excuse me. That's enough of those compounds to kill a horse. So that's not great, right? It's not especially useful to have an assay that only detects the compounds 2 to 3 orders of magnitude higher than you need. So what can we do about that? Well, we turn to biology. So we can tell the cells now to do something, and we're going to start out by expressing a fluorescent protein, GFP, green fluorescent protein, in response to those degradation products. So all this is showing you is it's called a circuit diagram, and it just shows you the genetic circuit we programmed into the cells. So what we have here is what's called a transcriptional activator, just a protein that tells the cells to start making whatever this is. And in this case, it's green fluorescent protein. So we can see if the small molecule's there, we can see fluorescence by eye, right? Without it, we don't see much fluorescence. The problem is that was not an especially sensitive technique. So we turned to my favorite microbe, Shewanella onadensis. Shewanella onadensis, or Shewy, was originally isolated from pond scum in Lake Oneida, New York, hence the onadensis. Scientists are very clever. That was a joke, you can laugh. And what scientists found is that these isolates grew really weird biofilms. So anyone who's looked at microbes in a biofilm before, these are weird, right? We normally don't see these needle-like structures. Well, those are what allow Shewanella to do this really cool thing called extracellular electron transfer. So we breathe oxygen. We put electrons onto oxygen, right, for respiration. These microbes, if there's no oxygen around, can actually spit out electrons outside of their cell onto things like metal oxides in nature or in the lab onto electrodes. Cool, right? Yeah. So that's basically how they respire. So they can take carbon products in the water, turn those into CO2, take those electrons, and move them extracellularly instead of reducing oxygen. So the way those work in practice for us is we can put them into things like a fuel cell and actually look at current growing in. So this is the cells colonizing the electrode, so growing a biofilm on an electrode and actually generating current directly. And the way they do this, so this is the inner membrane and outer membrane of the microbe, is with a series of proteins. And all you need to know about these proteins is that they have enough heme in them, so basically enough iron that they can act like a wire. So this basically allows electrons to flow directly from the inside of the cell to the outside, which is not something that normally happens in biology, right? But we can now take this circuit and transform it into Shewanella. So we can move it from, this was an E. coli, now we can put it in Shewanella. And so now instead of making this green fluorescent protein, we're making this SIM-A. So we can knock out that protein in Shewanella and prevent those cells from generating current. We can then what's called complement that protein back in, which just means put it in on a plasmid under specific control. So now what we see is if we have that small molecule present, that p-nitrophenol, we see current turning on. And we only see current in the presence of that p-nitrophenol. Make sense? So can we put these together? Unsurprisingly, E. coli grow under very different conditions than Shewanella, so it can make it very hard to try to grow them together. The great thing for our system is we've already killed these E. coli, so we don't need to keep them alive. So what we see here is if we throw these Shewanella or these E. coli in with these engineered Shewanella, we see charge, which is basically just integrating under the current curve that I showed before, only in the presence of both types of cells and that pesticide. We get a little bit of what's called leaky expression or just kind of off-target expression if we have just the Shewanella with that pesticide, but we don't really see very much background here. And what was even more surprising to us is this is quantitative. Based on the amount of pesticide we put in, we see a proportional increase in the overall charge. And what that means is not only can we tell you whether or not there was paraoxin or parathion present, those pesticides, but we can tell you how much there was. And our limit of detection here, when we calculate out, is 500 nanomolar. If you remember from the UVVis, it was about 5 to 20 micromolar. So we are 2 orders of magnitude below the limit of detection with UVVis in a device that costs us about $5. So what we've been able to do by engineering 2 individual strains of microbes is to both degrade these pesticides and detect the degradation products. And we can outperform much more expensive and difficult to use equipment. So why does this matter? Well, what we're doing now is putting this into masks so that we have disposable masks that contain liners that have these microbes and small sensors that have these. So farm workers for several pennies can protect themselves. Questions on this story? Still one more story to go. Yeah. That's a great question. So we can actually keep Shionella alive for a pretty long time, months, by embedding them into agar. So you guys have seen agar plates. That's what you grow microbes on. We can embed them into that. And as long as it stays moist, which it can with general humidity from the atmosphere, they'll stay alive for a few months. That's a great question. That's a great question. So that is more of a fundamental bio question. And basically, so we set the electrode at a certain potential. So it's at a slightly oxidizing potential. So the electrons tend to want to go there from the biofilm anyway. And then the Shionella can either directly transfer electrons if they're touching the electrode, or they can secrete small molecules that have these, that basically are carriers for the electrons that unfortunately is just random chance. If they bump into the electrode, they will transfer their electrons. If not, they usually will transfer them to something in solution, whether it's either salt or water molecule or something like that.
I have a couple questions from online.
Yep.
So Rohan Dugad, do you want to ask your question?
Yeah, so I might have missed something, but to my understanding, this will like the current is proportional to like the level of pesticides, right? So then by putting it in the sensor, the sensor in the farmer's masks, it'll like tell them how much harm they're susceptible to, but it doesn't actually like neutralize the pesticides, right? Like it just kind of gives.
Yeah, that's a great question. So the question was basically the Shionella detects the pesticide, but it doesn't neutralize it. So the great thing is the first microbe, the E. coli, actually does neutralize it. And because they're separate microbes, we don't have to use them into 1-to-1 ratio. So we can load that mask full of those microbes that can degrade the pesticide, which actually happens much faster than what I showed. From our experience, if we put enough microbes in there, it happens basically faster than we can measure. And then we can detect those degradation products basically just to let the farmer know if they've been exposed. So the goal of the detection is simply to let them know if they were exposed without actually having them be exposed, right? The idea would be the mask would degrade everything beforehand.
Okay, thank you.
And Meryl Zhang, you had a question?
Oh, I was just responding to like the initial question by saying like it can detect PNP and that's like the product of when they're already degraded.
Yeah, exactly. Mm-hmm. Yeah. It can, that's a great question. So the question was, can the pesticide get in through your eyes? So it can, the main route of exposure is inhalation because it targets muscles mostly. So it's mostly paralyzes your lungs. Skin exposure is not as big of a deal. And so mucous membranes are also not as big of a deal. Yeah, it's a good question, yeah. That's a great question. So do the pesticides still harm you if you eat the food? And no, at least not to our ability to detect it. So basically these guys degrade pretty readily. Those organophosphates degrade readily in under UV exposure. So they're really only active for killing pests when they're aerosolized. So during the first spraying. So if we can protect farmers during the spraying, we can pretty much protect them. So yes, generally they try to limit the spraying. That's also for cost. They don't want to spray where they don't need to. So unless there's like huge winds, which normally they will predict before spraying, it's less of an issue for exposure. Although I will say some places use this for mosquito control. So if you're in a suburban area and they're spraying for mosquitoes, this is probably what they're using. So don't go outside while they're spraying. You would think that, but basically it balances with them oxidizing usually organics in the water. So anything containing carbon in the water, they turn into CO2 and take the electrons out of that. And then at the end of their respiratory chain, they spit out the electrons. So it basically, if they didn't spit them out, there would be overly negative charge inside the cell. So they only basically release charge when they need to balance it. It's honestly mostly either agricultural workers or people who are exposed because of agricultural use or mosquito control. So generally people are exposed either because they're outside during spraying or because they're downwind of a spraying event. So it's generally people in developing nations who don't have either proper ventilation inside their homes or live directly adjacent to fields. That is I will say, much more esoteric question. So but the question was, can we engineer things like Shionella to work for things like a computer? The answer fundamentally is yes. Generally, our limit right now is how much charge each cell produces. So our limit of patterning, because these microbes are about 1 micron by 2 micron. So can we pattern them with enough precision to actually get circuits is the main challenge right now. But people are working on that. So post-exposure, if it's acute, which just means like one really large dose, you will have some permanent nerve damage, but you can flush your system and get rid of some of it. You can also use chelating agents that can pull some of it out of your nerves, but generally it's pretty much irreversible. That's why they use this mechanism for chemical warfare agents. Yeah, not
I have a host of questions.
okay.
Ash K. Manchanda.
Hi, thank you so much for the lecture today. I may have missed this, but I was wondering if there are any new measures being taken to protect farmers now that we know that long-term exposure is very harmful to their health.
Nope. Unfortunately, people who want to make money off of agriculture tend to not really care about farmer protection.
Rohit Kanamemi.
Yeah, so I just had a question about like how Shionella could be like maybe used for bioremediation since it can like reduce toxic like metals or
Yes.
substances. So like, is that like possible?
Yeah, definitely. People have actually been using things like Shionella and other microbes that can do this mechanism of extracellular electron transfer. People have used them for nuclear waste remediation. So there's a similar organism called geobacter that people use to turn uranium oxide back into soluble uranium, which makes it easier to remediate. People have thought about this a lot because there's a lot of things that are toxic in one redox state that are not in another. And so if we can use microbes to control the redox state of things in the environment, we can control their toxicity. That's a great question.
And just one more. Does the mask have some sort of visual apparatus that they can easily see how much they've been exposed to?
Yeah, it would basically be the equivalent of it's a reader that looks like a glucose sensor. So, yep. Yeah, they literally took a scoop of whatever was in that scum at the bottom of that lake and sequenced it and found Shionella. Cool, right? Awesome. So last story, we're going to talk about biological Velcro. But before that, I wanted to introduce the people working on it. So this is Dr. Gong Fan, a postdoc in my group who will be starting his own lab at University of Rochester in January. So if you're thinking about undergrad or grad school, you should look at his group. This is Yan, who is a sophomore right now in our group, and Juliet and Chao-Chi, who are first-year grad students. So carbon dioxide is bad, right? I don't have to tell this group. We can all agree on that. Excellent. Most of it is generated by human activity. What we don't often think about is that in theory, it could be used to generate valuable feedstocks, right? So if we add electrons fundamentally, obviously there's more complexity to it than that. But if we add electrons to carbon dioxide, we can make everything from methane to formic acid to methanol. And you can see that the prices of these per ton are very high. Feedstock is basically, that's a great question. So the question was, what's a feedstock? A feedstock is basically any sort of chemical you can use to make even more expensive chemicals. So CO2 could be a carbon feedstock, basically a carbon source to make all of these. And then these are considered feedstock chemicals because you usually don't use them in that form. You usually take them and make even more expensive chemicals out of them. Make sense? Awesome. So that's great, right? Why aren't we doing this with all the CO2 in our atmosphere? And this is just another market price conversion chart. Many of you will probably notice ethylene. That's the start of polyethylene plastic. So these are good starting materials for things that are called tough to decarbonize industries, things that are challenging to remove the CO2 production from. So why aren't we doing this right now? Well, right now it's very energy intensive to turn CO2 into anything else and usually ends up with more environmental harm than good. What my lab does, we use electrochemistry. So one of the ways we can take electrochemistry and harness it is by taking things like CO2 and water, which many of you will think of as seawater, right? The ocean is one of the largest sinks for CO2 globally. We can combine that with clean energy inputs, things like solar or wind, and use electrochemistry to convert that CO2 into those chemicals and fuels. That's great conceptually, right? The problem is the amount of energy we have to put in to convert carbon dioxide to carbon monoxide, the first step in that process, is very similar to the amount of energy it takes to split water. So if we're putting that energy into a water-based system, we're going to be generating a lot of hydrogen and not a lot of carbon monoxide. And that's not great, right? Because then all the energy we're putting in is going to targets we don't care about. And we can see that here. This is just basically showing you this is the potential of hydrogen evolution, and this is where CO2 reduction happens. So hoping by now you realize we will probably take inspiration from nature. This is an enzyme active site with what's called a heme in it, and I mentioned that earlier, but basically a heme is just an active site that has a ligand structure, which is just a fancy word for stuff around a metal center, and a metal center, in this case an iron. And so we took that ligand structure, which is called a porphyrin. Have people heard of porphyrins before? Heme, human. Okay, so heme is what carries oxygen in your blood. It can also be a catalytic site. That's what's shown here. We can also take that same core structure. So we see this structure looks a lot like that, right? So we can take this structure, and instead of shoving an iron in there, we can shove a cobalt, which is just a different metal center. And here, we can also make that same ligand structure with an iron. So these are just the three catalysts we're going to use. And they kind of look like enzyme active sites. So we can see here, we're going to talk about heme, cobalt TCPP, and that's just a fancy word for all the stuff around the cobalt, and iron TCPP. When we look at catalysis, we can look electrochemically. This is cyclic voltammetry, where we're sweeping across different potentials. And we're looking here at minus 1.4. We see without CO2, we have lower current. With CO2, we have higher negative current. That's great, right? We can quantify that. That indicates that there's catalysis, but usually with catalysis, we want orders of magnitude higher, not like 1.5x, right? So that's not great. Also, these catalysts are really unstable. So at the potential where we can actually do CO2 reduction, minus 1.4 volts, we see this negative increase in current. That's that metal center popping out of that catalyst and plating onto our electrode. So it's killing both the catalyst and the electrode. Not great, right? In fact, that iron version is so unstable, we can't even measure it immediately. We have to start at a lower potential and then move to a higher potential in order to see the degradation. This was not very fun for my postdoc when he figured this out. These catalysts have been known for about 30 years, and this is kind of an open secret in the field that they can do CO2 reduction, but they're pretty bad at it. So we wanted to see if we could use DNA to improve catalysis. So far I've talked about DNA in the context of engineering microbes, right? But DNA also has a three-dimensional structure. So in my group, we also think of DNA like a molecular Velcro. So if we think of the three-dimensional structure of DNA, it's two complementary sequences that bond together through hydrogen bonding, kind of like the electrostatics that keep two pieces of Velcro together, right? So if we modify one side of a piece of DNA with one thing and the complement with another, we can basically control their interactions based on tuning that sequence of DNA. So what we did here is we took one sequence of DNA and stuck that catalyst on the end of it. We modified our electrode with a complementary sequence. When we add them together, the hybridization efficiency is about 100%. So for every sequence on our surface, we're getting a complement with the catalyst. And so what we saw immediately was if we just take that free catalyst, if you remember, this was that instability. Now, if we just modify it with DNA, even before it's attached to the electrode, we see that it's much more stable, even at much higher potentials, down to minus 1.5 volts. So we've increased the stability. Not only that, but we can improve the overpotential that we need to do CO2 reduction. So this is a lot of data here. These two light bars in each of these are the free catalyst. The dark bars are the DNA immobilized catalysts. What you'll notice is at very low potentials, minus 1.15 volts, which is where we don't normally see CO2 reduction, we get 80% of our Faradaic efficiency going for carbon monoxide. And Faradaic efficiency is just a fancy word for saying for every electron we put into the system, how many molecules of product we get out. So normally with the free catalyst, we get about 0.2 molecules per electron. Now we're getting basically 0.8, and the rest is hydrogen, which is fantastic, right? In contrast, excuse me, to minus 1.15 volts or minus 1.5 volts, where we know those catalysts aren't even very stable, we see the majority of the product is hydrogen. That's not great, right? We're putting most of our energy into making something we don't care about. So at 30% less energy, we get 4 times the amount of carbon monoxide just by adding DNA. Excuse me. So what we've been able to do just by using this simple biomolecule to stick stuff together is we've increased the stability. So we can see this is that degradation of the free catalyst. We get a little bit of degradation with that single strand of DNA, but when we scaffold it on an electrode surface and we stick it to an electrode, we see no degradation. We additionally get that increase in Faradaic efficiency that we just talked about. So now we get basically 80% of what's produced as carbon monoxide versus at with the free catalyst, we only get about 20%. And finally, we get 3 orders of magnitude. So this is a log scale. Sorry, there's no actual scale on that, but the turnover frequency is basically how many reactions your catalyst can do in a given time, in this case per second. Here we see basically 1,000 times faster catalysis at 30% lower energy input for that DNA scaffolded compared to the free catalyst. So just by adding this little biomolecule, we can improve the catalytic efficiency, the selectivity, and decrease the energy input required to turn CO2 into valuable chemicals. And that is what I will leave you with. This is my group. I'm happy to take other questions. And like I said, if anybody's interested in seeing the lab, I'm happy to do that. And yes, questions. Yes, that's a great question. So the question was, are there any safety measures with the carbon monoxide produced? So we actually do this all in a sealed reactor, and we capture all that gas because carbon monoxide with a little bit of hydrogen is what's called syngas, which is an industrial starting material for a lot of other things people care about, like ethylene. So we want to capture it all, not just for safety, but also so we can turn it into things. That's a good question. Yeah. Mm-hmm. So we do, so the question was, we're not actually using the information in the DNA, just the structure. Yes, the only thing we use kind of the sequence of DNA to control is the melting temperature, because we can actually regenerate our electrodes by adding hot water, removing the catalyst, and then adding new single-stranded DNA catalyst to our surface. So based on our techno-economics of this, we think we're there. So by decreasing the amount of energy by about 30% and producing about 4 times the amount of carbon monoxide on electrodes that we can regenerate, so that on electrodes that we don't have to throw out once the catalyst dies, we think we're there. And we're now trying to put these into like shoebox size electrolyzers to make things like ethylene, to like swap out the DNA on there. 15 minutes. You basically add some hot water or buffer to it. It's called dehybridization. You break those hydrogen bonds, and then you add new DNA and let it cool down slowly, and it anneals or forms those hydrogen bonds again. Yeah, and it's the same. This is scale independent. So this is just basically how long it takes each specific DNA to attach, and we can do that at whatever scale we want right now.
I have a couple questions too.
Yep.
So since big agriculture isn't concerned about the farmers, where's the funding come from for all this research? Is it colleges and other programs like that, nonprofits?
Yes, that's a great question. So we get some of it from NGOs, which are basically nonprofits that support kind of human relevant missions. We also get some of it. MIT has some great internal funding from groups that are trying to ensure safe access to food and water, specifically the JWAFs group here. Awesome if you're interested in that. They fund a lot of really cool projects across campus. And then honestly, the military, so DoD is very interested in protecting people in general, but also soldiers. And because this does have relevance to things that look like chemical warfare agents, they're very interested in protecting soldiers from those.
Roha Dugad, do you want to ask your question?
Yeah, thanks for the great lecture. I was just wondering that since you said that you used DNA as like molecular Velcro, did it matter like which like species, I guess, or what kind of like DNA you used, or and like what did you like end up using?
That's a great question. So we actually chemically synthesize the DNA. Actually, we don't. We buy it from a company that does this it's about a couple pennies per base pair, and we use about 20 base pairs. So it's very short pieces of DNA. And the great thing is they can put whatever handles on it you want. So that's how we actually attach the catalyst to it, is we can put a chemical thing on the end that we can attach our catalyst to.
Oh, so you didn't like take like a certain species DNA or anything?
Nope.
Okay. Thank you.
Joshua Bello, do you want to ask your question?
Yeah, so I was just like trying to get more clear on the fact that it's like the DNA snapping together like Velcro that allows the reaction with the catalyst to take place.
So it's not specifically the DNA hybridization, but it's how close the catalyst is to the electrode. So for the reaction to happen, the catalyst has to be close to the electrode, but also close to a molecule of CO2. And so we need kind of all three of those things to be in proximity for that reaction to occur. And we think the DNA gives the catalyst enough kind of flexibility on the surface that it can react with CO2, but also be close enough to the surface that it's still reacting with the electrode.
Thank you.
Yeah, so that's a great question. The question was, can we do this for other types of emissions? We're actually very actively working on methane right now, because methane is much easier to reduce to ethylene than it is to upconvert CO2 all the way to ethylene. And we're really interested in ethylene for things like jet fuel and plastic production. That's a great question. So the question was, do we see logistical challenges with actually distributing our tech to developing areas? And that's something we actively work on. So I didn't show data here, but we do a lot of work on making sure that these proteins and things like that for the diagnostics are stable at room temperature, dried on our chip. So it's, not the most exciting work, but it's important. And we can actually keep all of the components of this stable, dried on a microfluidic device at up to 50 degrees Celsius. So we don't need cold chain transport. That's about 125 Fahrenheit. So you can transport and store this globally where you need to without requiring cold chain. It's a really good question.
So we're going to have that be the last question. But if you have more questions, we can certainly get them to Ariel. So make sure you send them along.
And I can hang out for a few more minutes afterwards.
And yes, so if your TAs are in a rush, we can walk you back to your class. So Sophia Ishan.
Oh, thank you. And I think
Hold it up
yes.
on the other side of her. Sure. So everybody, thanks to Ariel. Give her a big round of applause.